scientists have uncovered a miraculous process happening in each and every one of you. It's called the cell cycle. Let's go to a reporter live on the scene with scientists Dylan McCarthy and Alicia Theologides Rodriguez to hear more about this special phenomenon.
Thank you, Zoe. I'm here at Sage Hill School where the scientists are hard at work. Let's see what they're up to.
Hi, this is Dr. Alicia Theologides, and I'm her assistant, Dylan McCarthy, and we're going to explain the cell cycle.
So the cell cycle is made up of two parts. There's interphase and reproduction. Interphase includes G1, synthesis, and G2, and reproduction is all in mitosis. G1 is the longest of the phases, and they get shorter and shorter as you go on. However, not all cells are in the cell cycle. Some of them don't even reproduce, and those that don't are in G0. Now, not all cells are in each cycle all the time. There are cells that are always in reproduction, which include cells in your digestive tract, and then there are cells that are always in G0, which include those of neurons that stay with you forever and don't reproduce. And there are ones that switch over from time to time depending on whether they need to reproduce or not, and that includes the skin cells.
So let's dive a little deeper into the G1 phase. The G1 phase, like Dr. Alicia Theologides said, lasts the longest. It's about 11 hours. In this phase, protein and organelle synthesis occur, and the cell grows. Normal cell functions also occur in this phase.
So here we have the cell growing, and more organelles like the mitochondria and the lysosomes being produced.
Then we move on to the S phase. S stands for synthesis. This is about 8 hours, and this is where the DNA is replicated. There is limited organelle synthesis, and each chromosome is copied. So at the end of the S phase, there are 2 copies of the DNA.
Now let's talk about G2. In G2, it's 4 hours long, and protein and organelle synthesis still occurs. There is normal cell function, and the biggest difference between G2 and G1 is not only that it's shorter, but that the chromosomes are now, there's twice as many chromosomes, the centrosomes duplicate, and spindle fibers begin to produce.
Let's jump into mitosis.
Now we're in mitosis, which starts with prophase. In prophase, we have all the DNA in the center here. And the most important thing that happens in prophase is the breaking down of the nucleus. Now that the nucleus is broken down, it is really easy to recognize metaphase because all of the chromosomes line up in the center. Then the centrosomes, which were duplicated in G2, move to the ends of the cell, and they produce the spindle fiber, which attaches to the kinetochores on each of the chromosomes to pull apart the sister chromatids. Now in anaphase, we see each of the sister chromatids beginning to pull apart while the spindle fibers are still connected to them. At the same time, there are polar spindle fibers that reach from pole to pole on the cell, and their job is to push apart the cell to make it grow bigger. Now, in telophase, new nuclei form, and the chromosomes go into each. The centrosomes split apart in each side, so we have one copy here and one copy here. And to eventually perform cytokinesis, which is the separation of the cells, we have a contractile ring here in between them, which is made up of actin filament, which will eventually contract, so we will make 2 new daughter cells.
Thank you so much, Alicia and Dylan. I learned so much about the cell.
No problem.
Back to you, Zoe.